Hello, everyone. Welcome to this lecture. So the title of this lecture is Basic Science Update, New Target and Options for Pain Treatment. My name is Paulo Branco, and I am an assistant professor of anesthesiology and pain medicine here at Northwestern University. I have a PhD in neuroscience, and in my research, I study how the brain gives rise to pain perception. And I'm specifically trying to understand what are some of the risk factors that make an individual develop chronic pain after an injury. My email is also here, so feel free to reach out if you have any questions or if you want to discuss some of the topics of this lecture. So I don't really have any conflicts of interest to declare. And before I actually start the lecture, I kind of want to start by raising a few points about the material that I will present today, and also acknowledge some biases in the material that I selected to discuss with you. So the first thing that I want to discuss is that if you notice, the two parts of the title of this talk stand a bit in contradiction. From the research timeline, basic science and then the treatment are usually on two polar opposites, and most discoveries from basic science take years, if not really decades, to be approved as treatments. So for this talk, I opted to select a few examples from the literature that showcase some relatively advanced projects that stemmed from basic science and are on the way to be tested for treatment efficacy. Now, I want you to keep in mind that none of the things that we will discuss have been approved or should be taken as a fact, because, as I'm sure you know, science requires proper scrutiny and replication, and we need to give it some time until we can take, again, these findings as a fact. So the second thing that I wanted to acknowledge is that in order to make this lecture appealing to physicians, I will discuss mostly findings that follow what we call the translational and reverse translational approach. So what this means, and I try to kind of represent this on this figure on the right, is that we first try to understand the human condition that we want to treat, and then we go back to animal models, preclinical models, to actually tap into the mechanisms using basic science, test new interventions, and then try them again on humans once we know exactly what mechanisms to tap. So while there are thousands of basic studies that we could discuss, it makes more sense that we discuss basic science that has been inspired by human research. Finally, I also want to acknowledge my bias, especially because I'm a neuroscientist, that pain is in the brain, and therefore, with few exceptions, I will focus on discussing the role of the brain in pain. So I also want to tell you that this talk goes hand in hand with two different talks from two of my colleagues, namely the lecture number 40, From Synapses to Consciousness, Understanding Pain, and lecture number 43, Imaging Research to Clinical Care, Advancing Pain Treatment Options. We cover some similar topics, but from different angles. And so I encourage you to listen to them after this lecture so that you get a full picture of the kind of things we're going to be discussing. So before we even turn to discussing pain, I first want to start by discussing perception. So this example here on the left was quite famous a couple of years ago, I think about 10 years ago or something now. And it has become very popular because there are some people that actually see this dress as white and gold, while others say it's actually blue and black. So I'm actually on the blue and black side here. And to be honest, I just cannot possibly imagine how someone can see white and gold on this picture. But there is actually an objective answer to this dilemma. So the dress is actually black and blue because people actually went looking for this specific dress. And in fact, if I take this same figure, the same image, and I pass it through any image editing software, you can clearly see that the colors that are contained in the image are basically shades of blue and shades of brown. And this distortion in some of the colors is caused by the fact that this picture is overexposed. And so again, the colors are slightly distorted. Now, what is actually interesting in this phenomena is that a bunch of studies have studied this. And the conclusions are not particularly earth-shattering, but we kind of learn a couple things about perception from looking at this example. So this is one such example of a study published in 2017, where they looked at about 13,000 individuals who had basically to decide whether the dress was blue and black or white and gold. And what they found was that about 2/3 of the subjects would rate this dress as being white and gold, and then a third blue and black, and then there was a residual number of subjects that reported a combination of both. Now, the interesting thing about this is that the researchers actually try to understand why do some people think that this dress is white and gold, while others think that this is black and blue. So they asked a bunch of questions to the subjects. And I will actually tell you the answer right away. And it seems that one of the strongest reasons why some people believe it's either, again, black or blue or white and golden has to do with how you assume that this picture was taken. So basically, if you believe that this dress was exposed to direct light, you will think that you are much more likely to see it as black and blue. And if you think that the image is in shadow, you are more likely to report that it is indeed white and gold. But this interesting phenomena doesn't really stop here, because then they actually looked at a bunch of other parameters that could potentially explain why you would even assume that the dress is in shadow or exposed to direct light. And what they found was that there are individual differences that explain your perception of the dress. For instance, it seems that females are more likely to believe that the image was white and golden here compared to males. They also found that, for instance, age plays a role, although I'm not showing this on these slides. But to me, the most interesting result is that your circadian rhythm, or maybe perhaps more specifically, whether you tend to be a morning person or a night owl, also impacts how you see this dress. So this is speculated to be a consequence of your exposure to light, which makes intuitive sense. So if you tend to see a lot of direct sunlight because you wake up early and you see a lot of direct sunlight, then your brain is a little more tuned to images that are overexposed, such as this one. On the other hand, if you are a night owl, you are more exposed to artificial lights, and so your brain has experiences with different patterns of lightning. And so this in turn creates what we call the Bayesian prior, which means that your brain, given its experiences in memories, will interpret the same objective sensory stimuli in a very unique way that depends again on your experiences and your memories. There has even been studies looking at this phenomena of the dress, looking at how different brain regions might explain differences between individuals in how they perceive, again, the color of the dress. So you might be asking, why did I go through the trouble of discussing a social media trend in the context of this presentation? And the reason is because I want to start this lecture by addressing the fact that pain is in fact a subjective experience that builds on top of sensory stimuli that arrives from your body to the brain. And this is what generates the pain experience that we are all familiar with. So it is probably not very strange to believe that your brain may be able to amplify or even misinterpret bodily signals, and as a consequence, generate a heightened state of pain. So in this case, perhaps we can develop ways to help the brain interpret these bodily signals in a more adaptive way, and maybe lead to new treatments that help us minimize the burden of pain. So the concept that pain is a subjective experience is not particularly novel. Indeed, if you take a quick look at the definitions from the International Association of the Study of Pain, they actually define nociception as the neural processes of encoding noxious stimuli, and they also state that pain sensation is not necessarily implied. If we go to the figure on the right, you can see that we have these highly specialized neurons that detect nociceptive stimuli, so they are called nociceptors, which convey this information to the spinal cord, where the first level of processing takes place. And then this information is relayed to the brain, where pain is actually generated. So the reason why pain sensation is not necessarily implied upon the detection of noxious stimuli is that the brain actually interprets these nociceptive inputs and then generates the pain experience. So as you can see from the definition from the International Association for the Study of Pain, they describe pain as an unpleasant sensory and emotional experience associated with actual or potential tissue damage, or described in terms of such damage. So this definition implies that there is indeed some level of subjectivity in the experience, and also highlights that pain is a multifactorial experience that likely depends on several factors. We will in fact discuss some of these factors in a couple of slides. Still, to reiterate, the important takeaway message here is that the pain that someone experiences is not linearly related to nociception. There are several examples of phenomena that clearly show that you can modulate pain perception without changes in the sensory input itself. Now, two of my favorite ones are the placebo and the nocebo effects. So the placebo effect is caused by positive expectations, where basically you expect to feel analgesia because you think that you're receiving an active compound, and as a consequence, you're actually going to be perceiving less pain. Now, the dark side of the placebo effect is a nocebo effect, where if a patient has negative expectations about a given outcome, they are more likely to report higher pain intensity or even report negative side effects. Now, since both of these phenomena, you are not actually changing anything in the periphery. So basically, they need to be acting towards the brain, through the brain. So there are a few fun anecdotal examples of these phenomena. And here on the left, you actually see a case report of a construction worker who fell and stepped on a nail. So as you can see here on the picture, the nail actually pierced through the boot. And in this case report, they actually mentioned that the patient went to the ER complaining of excruciating pain. But when they actually removed the boot, they realized that the nail had passed between the toes. And if there was indeed any injury, it was probably nothing much more than a scratch. Now, I'm obviously kind of speculating here, but it's likely that this vivid realization of having a nail go through your foot made this patient much more susceptible to perceiving these minor noxious stimuli as something that could be life-threatening. There is another fun example that I have here on the right that was actually reported in several news outlets. So this is a subject that accidentally discharged a nail gun, and the nail went through his cranium, as you can see here. Now, I probably don't need to tell you about this, but this looks pretty painful. But the patient actually reported, when he went to the doctor, he actually reported this as a minor inconvenience. And only later, he actually started to describe what was a minor toothache. When he actually went to see the doctor, it turned out that this was a 4-inch nail. Now, these are some extreme cases, probably also boosted by some news sensationalism. But as you'll see next, this has actually a very strong correlate with how we see pain in the clinic. So this is a bit of a busy slide, but bear with me. So what I'm showing you here on the left is a systematic literary review published in 2015, which attempted to quantify the percentage of subjects who, while having no pain, so these are asymptomatic patients, show imaging features that would be categorized as spinal degeneration. This is actually a pretty big study with a lot of subjects and a lot of studies. So if you look at a certain age, so let's look at 30 years old, which is quite familiar to me, you'll see that about half of the subjects, again, completely asymptomatic patients, show imaging findings of disc degeneration. If you fast forward to age 50, you will see that about 80% of individuals have at least some form of spinal degeneration. Even other findings like disc bulges, protrusions, facet degenerations are observed at strikingly high levels. So the conclusion of this study is that basically incidental findings of spinal degeneration are not by themselves really indicative of whether someone is in pain or not. Now, of course, you need to interpret imaging findings together with the clinical presentation. Still, this clearly shows that perhaps in some of these individuals that don't feel any pain, maybe nociceptors are still detecting the injury, and for reasons that we will discuss in the future in a couple slides, this is not really brought to the brain's attention. Now, on the right, we're looking at another study, but now this study is actually looking at patients with chronic low back pain and also pain-free subjects. So they actually performed an MRI to, again, quantify the number of findings observed in patients and see if basically the prevalence of findings is the same or not in patients with chronic back pain or healthy subjects. By healthy, I mean patients with no pain. So what you find on the graph below is that, so this graph here, is that there is not a substantial difference between subjects with and without pain when it comes to these MRI findings. I want obviously to add some nuance and say that this depends on the type of imaging sequence and even the clinician, but I would personally expect a much larger difference between the two groups. However, this data doesn't really match these expectations. Now, this study also did something interesting. They looked at the patients that had no pain and tried to understand if, based on the number of imaging findings, they could predict whether they would develop chronic pain in the future. And I think the future here was either 6 months or 1 year. I don't remember exactly. But what they found was that indeed the number of imaging findings was not a strong predictor of whether they would develop chronic pain. The only really significant finding was that patients with no pain but with 4 or 5 or more findings had a slightly higher likelihood of developing chronic pain in the future. So again, this really highlights how these body degeneration or body injury doesn't really relate with the amount of pain that a given individual reports. So I want to be careful and also mention that the brain is not the only player in pain, although it does generate it. So the brain does interpret the nociceptive information being provided by your peripheral and central nervous systems. And we know that as patients develop chronic pain, there are a lot of changes occurring at multiple levels within both nociceptive and pain processing pathways. Now, we won't have time to discuss these at a great depth, but I still wanted to include this slide to show you that we know quite well that, for instance, nociceptors can become sensitized over time and fire in response to stimuli that would otherwise not be detected as noxious, or maybe in other words, subthreshold stimuli. We also know that inflammatory processes caused by the injury play a role in sensitizing nociceptors. There's even some evidence that some of these inflammatory processes can reach the spinal cord and even the brain. This is often described, by the way, as peripheral and central sensitization. We're going to talk about this in a couple of slides. And this is a topic that I will cover very briefly, mostly for time issues, but I do recommend, if you're curious, to read this review by Meacham and colleagues that explains this quite well. So regardless of all of this, it is generally thought that these changes that occur at the periphery and at the spinal cord act as an amplifier. So they kind of turn the quote-unquote volume of the nociception up or down. So where this actually gets interesting is when we think about the role of the brain and how it does interpret these nociceptive signals that are arriving to the brain. So there are several theories about this, and I am partial to a theory developed here at Northwestern by Vanya Apkarian, who happened to be my mentor. This theory suggests that the brain acts as a dynamic filter, allowing a given nociceptive information to rise to perception if it passes a certain threshold. And this threshold is decided based on the motivational value of the environment. So in other words, your brain at any point makes a direct assessment of whether a given incoming nociceptive input should be brought or not to perception, given some biological imperatives, like, for instance, if you are in a threatening situation, or if you are depressed, or if you have like a very aversive experience and things like that. So we are going to discuss this model also in a couple of slides, but for now I want to switch focus to another concept, which is actually the last point that I'm going to make in this short introduction before we get to the science. So as I mentioned before, acute pain is kind of advantageous for us because it allows us to kind of tune our behavior based on the biological and the environment imperatives. So if you're in a very stressful situation, it's actually quite advantageous for you to either feel pain or not feel pain based on how that would impact your survival chances. But it is also important to note that the majority of individuals that experience pain will eventually recover. It's only in some cases that pain might persist beyond tissue healing. And maybe here I should clarify that I'm talking about conditions where there is no clear underlying reason for patients to feel such high levels of pain. So, for instance, examples like chronic cancer pain, while the same principles apply, the source of nociception in this context will be more easily identifiable. Now, the concept that I want to discuss in this slide is the transition from an acute stage to a chronic pain stage, which allows us as scientists to try to understand what happens both in the periphery, but also in the central nervous system that seem to perpetuate these painful states. So I want to separate this transition into three different stages. So first, we need to acknowledge that there are some predisposing factors that might increase the risk of developing chronic pain. A few have been identified, for instance, immune status. My mouse is here, my immune status. Again, it has been shown that, for instance, people with adverse childhood events and who, as a consequence, have been exposed to chronic stress have exaggerated immune responses to a given stressor. And so they are more likely to develop chronic pain after a given trigger. Like sex, genetics, and age have also been identified as risk factors. And then there's also a bunch of psychological aspects such as anxiety, depression, pain catastrophizing, which are also thought to play a role, even though we don't really understand why. Now, of course, there's going to be a trigger for the pain, which typically is an injury. And of course, this also relates to the outcomes, mostly because of the mechanisms that caused the injury in the first place. But perhaps more interestingly, after the injury, over time, there are a series of physiological events that will take place and increase the risk for the development of chronic pain if they are not reversed. These include, as I mentioned very briefly before, peripheral sensitization, either caused by the sensitization of nociceptors or ongoing inflammation, sensitization of spinal cord, dorsal horn circuitry, and also several changes in the brain, which we will explore. Finally, there is a maintenance phase where these maladaptive changes that occur during the transition are not allowed to revert back to the normal state. And then as a consequence, they might maintain this chronic pain state and this vulnerability state that will enhance the chronic pain experience. So now we're going to switch to the science. And I did mention that inflammation responses might impart risk for the development of chronic pain. And here I actually want to highlight a recent study that was quite spoken about in the pain community following a reverse translational approach with both humans and animals. And for brevity, since we're already a bit delayed, I'm just going to say that I'm not really going to explain the mechanisms behind these inflammatory processes, but if you're curious, these two reviews do a very good job at explaining how inflammation can actually affect both the periphery, the spinal cord, and even the brain. So I wanted to share this study with you because it's relatively recent and it's been quite discussed in the pain community. And if true and replicated, it has very important implications for clinical practice. So this study was conducted by the group of Luda Diachenko from McGill University, and they collected a sample of about 100 patients with low back pain during an acute pain episode. So they defined this as basically recruiting these patients at least up to 6 weeks after pain onset. And then these patients actually returned for a follow-up visit about 3 months later. So on the left, you can see some examples of some pain trajectories. So some patients persisted in their pain, symbolized here in orange, while others actually recovered from their pain, which are here illustrated in green. So what they did in this study was to study genome-wide transcriptomics to examine whether gene expression differed between patients who eventually recovered versus those that developed chronic pain. And here I'm only showing you the pathway analysis that they report on this paper, which basically groups individual genes based into their functional roles. On the right here, you can actually see 4 different comparisons that they performed. So the first column is trying to ask the question if there are any differences between the patients that in the future either persisted in their pain or recovered, but when you are assessing them at this acute stage. So as you can see below, they actually found that patients who recovered had higher gene expression levels related to inflammatory responses, neuroinflammatory responses, and also acute inflammatory responses. So this suggests that, at least at the acute stage, it seems like these inflammatory responses might actually be protective against the development of chronic pain. Now, when looking at cell activation here in panel B, you also see a lot of gene expression related to the usual suspects, such as leukocytes, neutrophils, and macrophages that were substantially upregulated in the patients that went on to recover in the future. Now, the second column kind of asks the same question, but instead looks at the difference between the two groups, but now in the follow-up visit. And as you can see in the bottom, there are pretty much no differences between these two groups, suggesting that whatever happened in the first visit was a transitory phenomena that then reverted to normality. And then finally, in the last 2 panels, panel 3 and 4, they are actually looking at changes within subjects, so within each group, to see if there were, again, changes over time in the patients that either persisted in their pain, here in orange, or that recovered in their pain, here in green. And what they found was that for patients that persisted in their pain, there were very minor changes, but for patients that actually recovered from pain, they actually found, again, a very big change, so a notable decrease in all features that were overexpressed at this acute stage. So basically, all these 3 parameters are reduced after 3 months. So taken together, these findings in humans suggest that inflammatory responses in the acute stage seem to be important to potentially promote tissue healing. And if these responses seem to be suppressed, then individuals might be at a higher risk for developing chronic pain. Now, this evidence is mostly observational, and causality here is a bit difficult to demonstrate. But what we can do is we can turn to animal models, which the authors did, to try to understand this phenomena a little better. So what they did here is they induced chronic pain in mice using several methods. So the one that I'm showing in this slide is an inflammatory chronic pain model, but they also studied neuropathic pain with similar results. So what they did, this is called the CFA model, where they exposed the paw of the mouse to an inflammatory agent called the Freud adjuvant, creating significant inflammation that lasts for a few weeks. Since you can't ask mice how much pain they feel, the way that we typically assess their pain is by poking their paw with hair filaments that we call von Frey filaments, which basically apply a certain force that here in this study was measured in grams. So this force is increased until basically the mouse withdraws their paw, which we take as a proxy for pain sensitivity. The idea here is that the quicker they withdraw the paw, or basically the less force that you need to make them withdraw the paw, the more sensitized they are to their pain. There's also other very frequent chronic pain rodent models that we're going to be discussing. One of them is called the spared nerve injury model, which they also tested in this study, where instead what you do is you cut 2 out of 3 branches of the sciatic nerve, and so the mouse can still move, but he will develop chronic neuropathic pain. So in these experiments, they asked the following question. If we give mice an NSAID after the onset of pain, and as a consequence reduce inflammation, do these mice end up with worse long-term outcomes, reflecting kind of human subjects that we saw earlier? So here, as you can see on the left, basically the black line represents patients that received an NSAID. In this case, it was dexamethasone. And the white lines represents the mice that just received saline. And in the acute stage, as you can see, the NSAID is actually lower the pain sensitivity for the mice that received, again, NSAIDs. But then after some days, you will actually see that the pattern completely reverts, and it seems like NSAIDs actually make the recovery much worse for these mice, which again goes back to this concept that we were discussing in the human study. So of course, this could also be simply because NSAIDs are causing pain relief. And so to control for these pain relieving effects, the authors actually repeated the experiments using several non-NSAID analgesics, such as gabapentin, lidocaine, and morphine. And they also used diclofenac to replicate the previous results with different pharmacological agents. So as you can see here, the results are pretty much replicating the previous results. So basically all analgesic agents induce pain relief at the acute stage. But then the mice that received the NSAID, which are the mice here represented in green, were by far worse off after 20 days. And then to further drive this point home, the authors actually looked at a big data set called the UK Biobank, and they identified 98 subjects that had acute pain and that were followed longitudinally over 1 year. So half of these subjects reported no pain after 1 year, while the other half reported significant pain, and the authors actually looked at the medication usage during the acute stage to again see if it could predict the outcomes 1 year later. Now, this figure is a bit complicated, but I want to draw your attention to the rightmost panel, where the authors built a prediction model, including a couple of features like usage of NSAIDs at the acute stage, antidepressants, demographic variables like age and sex, and psychological distress. So in this data set, patients didn't really report pain intensity because, I'm going to reframe this. So in this data set, patients didn't actually report pain intensity, but they did report the number of pain sites, which the authors used as a control for pain itself. Because as you can imagine, patients with higher pain might also be the ones taking more NSAIDs. And what they found was that patients taking NSAIDs in the acute stage had almost double the likelihood of developing chronic pain 1 year later. And I have to say that this is quite striking. And although I'm not sure there is yet enough evidence to stop giving people NSAIDs after, for instance, a surgical procedure or an injury, this is still kind of like a developing story, but it's still quite striking to me. So at this point, I think we still need to understand the exact mechanisms a little better, but it is quite interesting that it seems like inflammation may play a key role in the development of chronic pain. Now, I want to very briefly touch on central sensitization, and as before, I'm not really gonna discuss this too much for time reasons. There's a couple reviews that I'm going to link later that will explain this a little better. So here I first kind of want to emphasize that it is very difficult to directly measure both central sensitization and peripheral sensitization in humans. What we typically do in research, which is also heavily used in clinical practice, is to employ tools that are presumed to measure some of these peripheral and spinal processes. Now, this approach is often referred to as quantitative sensory testing, and it has been extensively researched. The general concept involves using either pressure or heat to induce pain in specific ways that allows us to measure phenomena such as allodynia, which is a response to low-intensity, non-noxious stimulation, such as light touch or non-painful heat. But we can also measure pain thresholds to determine, for instance, pain sensitivity and tolerance. There are also a bunch more measures that look at dynamic properties, such as, for instance, wind-up. For example, something that we can do is apply a stimuli several times to assess how these stimuli at the same intensity can evoke progressively higher pain reports, and this is thought to serve as a good proxy for central sensitization. Another frequently used technique is called conditioned pain modulation, which measures whether a given painful stimuli is perceived as less painful when there is a concurrent painful stimulus in a remote body location. This is thought to reflect both spinal cord inhibition and also the brain's control over spinal cord circuitry, which we will discuss again later. Now, we have conducted actually some of these studies. So one of the studies that I'm highlighting here on the bottom right was a study comparing patients with chronic pain because of knee osteoarthritis and also healthy controls. And what we have found was that patients with osteoarthritis of the knee reported significantly higher levels of pain in response to a simple pinprick in the skin around the patella, suggesting secondary hyperalgesia. So they are much more sensitive to a noxious stimuli. But perhaps even more interesting is that in this study, we actually assessed these patients before and after total knee replacement. Our goal here was to try to understand whether central sensitization or peripheral sensitization could predict pain outcomes after total knee replacement. So in case you are unaware, about 11 to 34% of patients undergoing total knee replacement develop chronic post-surgical pain that can actually last for months or even years after surgery, long after the tissue heals. And one of the leading theories is that although the joint is now removed, the spinal cord becomes sensitized after years of receiving massive nociceptive inputs. And so even after the joint is gone, the spinal cord will continue to respond to non-noxious stimuli and generate a state of persistent pain. Now, returning to the study that I was just mentioning, as you can see here on panel A, before the surgery, patients experienced significant levels of pain, and this obviously decreased as time elapsed after the surgery, as was expected. But you can see that there's still a couple of subjects that 12 months after still report significant levels of pain. So what we have found in this study is that, again, secondary hyperalgesia measured through pinprick was actually predicting pain outcomes 1 year later quite significantly. And perhaps more importantly, we also assessed secondary hyperalgesia through pinprick stimulation 3 months after the surgery. And what we found was that patients whose hyperalgesia normalized after the surgery were the ones more likely to report less pain 1 year later, while patients reporting either the same or even higher hyperalgesia after the surgery were the ones that were likely to develop chronic post-surgical pain. So this kind of tells us that using these kind of tools, we can indeed predict outcomes in these subjects. Now, one of the reasons why we don't know a lot about the mechanisms of central sensitization in humans is because the technology to directly assess responses in spinal cord is still lacking a little bit. There are still a lot of methodological obstacles that we need to overcome. Here I'm just briefly showing one example of some work that is trying to address some of these challenges. You are actually looking at a specific MRI protocol that assesses spontaneous neuroactivity within the spinal cord, here at C5, C6, and C7. And what they do is they compare just brain activity, in this case, spinal cord activity, not brain, between patients with fibromyalgia and just healthy controls. And what they found was that fibromyalgia patients showed lower spontaneous activity within the dorsal horn of the spinal cord, here in red and orange. and higher spontaneous activity in the ventral portion of the spinal cord here in blue. And this finding could suggest reduced dorsal horn spontaneous activity, potentially highlighting some local inhibition of ascending nociceptive pathways, which aligns with what we would think would happen in central sensitization. So it is our hope that with improvements in methodology, especially with better scanners and better methods, we are maybe in the future able to actually quantify central sensitization using imaging techniques. So after looking a little bit at the spinal cord, we are now finally moving to the brain. And before we actually continue, I kind of want to spend some time for us to get familiarized with brain anatomy and function before we actually move on to the science. So as I mentioned in the beginning, once a noxious stimuli is detected by nociceptors, this information is sent to the spinal cord, which then relays it to the brain. Now, I'm sure you are aware that the brain is an incredibly complex organ, and it's quite hard to summarize. And to make sense of it in the context of pain, I've kind of chosen to discuss it in using a classic theory, the neuromatrix theory of pain by Ronald Melzack. This theory argues that the brain performs three key computations with the nociceptive information it receives, and it can be summarized into some domains. So the first domain here in green is called the sensory discriminative domain and includes brain regions such as the posterior insula and the primary and secondary sensory cortices. These brain areas are thought to encode sensory information about the noxious stimuli, including its location, its intensity, and are frequently activated whenever you deliver a painful stimulus to a subject. So the second domain here in red is the affective motivational domain, which encodes the emotional component of pain. And this is composed mostly by what we call the extended dopaminergic system in regions such as the ventral tegmental area, the amygdala, the nucleus accumbens, and the medial prefrontal cortex. This system provides this nociceptive signal with the emotional valence that makes pain feel unpleasant. It also involves several structures that can augment the pain experience and also encode it into memory. So if you recall my earlier slides, these are actually some of these brain regions that we believe that act as a dynamic filter for incoming nociceptive signals. They basically retrieve previous experiences, assess the motivational value of pain given a certain situation, and then reflect individual responsiveness to a given aversive stimuli, such again as noxious stimulation. So the third domain here in blue is the cognitive evaluative domain, which integrates the sensory and affective components of pain to generate a unified representation of pain in the brain. So it involves regions that are major hubs, including, for instance, the anterior insula and the anterior cingulate cortex. And because these areas are big hubs or big highways of the brain, they are well suited to basically integrate the information being processed at other brain regions. These regions are also thought to be quite important to allocate attention towards these incoming sensory information. So the final and fourth domain, here represented here in dark blue, which I'm calling the modulatory system, also known as the descending modulatory system, is primarily composed of areas such as the periaqueductal gray area and also the rostroventral medulla, which have direct projections to the dorsal horn of the spinal cord. So these projections can actually locally inhibit ascending transmission of nociceptive information. And importantly, most of these three domains actually project to these modulatory systems that then in turn project to the spinal cord. And this is basically a pathway that allows the brain to again control ascending nociception. So with brain imaging, one of the things that we can do is do a couple of really interesting experiments and try to understand how the brain actually processes pain. So this is actually just a simple example where we can, for instance, take a simple volunteer and using MRI compatible equipment, we can actually deliver painful stimuli and see how the brain reacts. So brain regions that are showing high metabolic activity after painful stimulation will light up in the scanner, and we can actually quantify these responses and map them into different brain regions. So for instance, the brain regions that I'm showing you here, so these images, are actually average activity from a meta-analysis of about 516 studies involving thousands of individuals. So these studies show that, for instance, when you apply an experimental painful stimuli to a given individual, these areas light up. They represent a lot of the regions that we just mentioned, such as the insula, the periaqueductal gray area here, the rostroventral medulla, the anterior cingulate, even the thalamus here. But you don't typically see affective brain regions in these experiments. And this is because these maps are collected in healthy subjects in lab conditions. So this affective component of pain is not very pronounced. So in a way, this type of pain that we typically do study in the lab is not very ecological because it's artificial and it's predictable. And so these affective regions are not overly active. However, with brain imaging, we can do some really interesting manipulations, trying to, again, assess different expectations that patients might have about how a given painful stimuli is going to be perceived. And this might give us clues about how pain is interpreted in the brain. So this is actually one of my favorite neuroimaging studies, and I think it highlights this quite well. So now that we know how a typical brain responds to pain, especially in healthy subjects, we can now start talking about what happens in the brain with chronic pain. So this study that I'm showing you here was one of the first to use neuroimaging to attempt to see how the brain changes as the pain transitions from a subacute stage to a chronic pain stage. And what they did was to recruit a large sample of subjects with low back pain about 1 to 2 months after pain onset. So again, at the subacute pain stage. So what they did is that they scanned these patients at that point, and then they also scanned the patients at multiple time points up to a year. So while the patients were being scanned, the patients were asked to rate their own pain so that we can actually measure the brain activity that is related to their pain. So again, these patients were followed over time, and based on their pain levels 1 year later, they were segregated in 2 groups. The first group, which is this line over here in black, shows patients whose pain persisted. And the second group shows patients that recovered from a subacute back pain. Now, I will say that the patients didn't completely recover, but they showed at least some improvement, and their pain was on average 2 out of 10, which is, or 20 out of 100, which is considered kind of mild pain. So since these patients were actually scanned multiple times throughout the transition to chronic pain, we can actually see how the brain changes as pain either resolves or persists. And what's interesting in this study is that for the patients whose pain recovered, which is here this bottom row, their brain activity initially looks very similar to the brain areas that we already established are active whenever you, for instance, induce pain in a subject. And then as pain recovers over time, then the signal kind of becomes harder and harder to detect in the brain. So in other words, the pain-related activity kind of disappears over time in the patients that recover. However, in the patients that actually persist in their pain, what you see is that in the first visit, their pain maps to these sensory areas that, again, we already established that are related to just normal acute painful stimuli. But then as time evolves, you will see that the brain areas that are being recruited to represent this pain are mostly these affective and motivational brain areas that code these affective aspects of pain. So in other words, what this study is showing is that as patients develop chronic pain, there are a bunch of plastic changes happening in the brain that will heighten this pain experience, this affective pain experience over time. So in this same longitudinal study that I just presented, the authors also asked a different question. Can we, based on brain properties from the first scan, so that is collected 1 or 2 months after pain onset, predict which patients are going to develop chronic pain 1 year later? So what the authors did was they looked at how different brain regions were communicating with each other and tried to see if, based on that, they could predict again whether patients were going to recover 1 year later or persist in their pain 1 year later. And what they found in this study is that the amount of information being exchanged between the nucleus accumbens and the ventromedial prefrontal cortex, again, 2 areas of this dopaminergic system, so the higher this connectivity, the more likely the patients were to persist in their chronic pain. And these actually obtained like some very significant areas under the curve. For instance, at 1 year, the AUC was 0.83, which is a quite significant value. So in other words, these areas which seem to be quite important for the effective evaluation of pain seem to be predicting who's going to develop chronic pain at the subacute pain stage. So in this slide, I want to kind of go back again to some basic science and highlight the importance of translational research. So here, what the authors did is they went back to the preclinical models. So this is a spare nerve injury model. So basically, again, we cut the sciatic nerve. We cut two out of three branches of the sciatic nerve. And what this does to the mice is that they can still move, but they feel neuropathic pain chronically. And because we are doing this in mice, now what we can do is we can sacrifice them and actually look at the neurons of, for instance, the nuclear accumbens, like we just looked in the slide before, to see if there are like plastic changes as the mice develops chronic pain and whether we can try to revert them. And indeed, in this study, what they found was that some specific neurons of the nuclear accumbens, namely the spiny projection neurons, seem to have increased excitability in the animals that had chronic pain, which are here in green called SNI animals, compared to sham animals. And the other finding that was very interesting was that the dendritic complexity in the chronic pain animals was lower, smaller, compared to sham surgery animals. And I didn't mention that, but the sham surgery is simply when you do the surgery, but you don't actually cut the nerve. So basically, these mice recover quite well after a while. Now, what is interesting in this study is that, again, because now we are trying to assess these mechanisms, we can actually try to revert them. And so what in this study they have done is they administered L-DOPA after the injury to increase dopamine availability in these regions, together with naproxen, as a way to reduce the painful stimuli and try to mitigate some of the nociception. So L-DOPA was administered here because, as I mentioned, both the nuclear accumbens and the medial prefrontal cortex are part of the dopaminergic system. And so it could be the case that L-DOPA could help normalize this system. And so here again, they take the mice, they perform an injury, and then they give L-DOPA and naproxen at the acute stage. And what they found is that they're actually able to normalize both the excitability of the nuclear accumbens neurons to similar levels of sham animals. And also, it also normalizes the dendritic complexity. So basically, it stops these changes in these neurons from taking place. And perhaps even more importantly, here on the right, you will see that the pain analgesia of these patients, which we measure again using von Frey thresholds. Again, we poke the mice with a von Frey filament to see how quickly or how much force is needed for the mice to withdraw the paw. And basically on the left, we are looking at sham surgery animals. And as you can see, they have pretty normal mechanical allodynia. So they withdraw their paw relatively normally. But then if you induce a spare nerve injury, then animals that receive saline, so they are not treated, they show very enhanced mechanical allodynia, which we take as a proxy for pain. But if you actually treat them with L-DOPA and naproxen, then these mechanical allodynia completely normalizes, again, showing that this treatment seems to block the development of mechanical allodynia. There has also been some studies with mice and rats where you can use optogenetics to actually directly stimulate some of these areas. So this study actually stimulated the prefrontal cortex. And when they stimulate, for instance, here, the prefrontal cortex, what they found is that in sham animals, it doesn't really make a difference because, again, these mice don't have a mechanical allodynia. They are perfectly normal. But in mice that had a spare nerve injury, when you actually activate the prefrontal cortex, you will see that their mechanical allodynia levels return to normality very quickly after. So again, showing that it's not just pharmacologically, so it's not just that we pharmacologically can block the transition to a chronic pain state, but if even in the chronic pain state, if we stimulate these areas, then these mice become arguably pain-free. Now, this concept was actually tried to be, I'm going to repeat this, trying to move too fast, and so I'm kind of stumbling a bit on my words. So you might think that some of these preclinical findings suggest that this could be a potential treatment to block the transition to chronic pain in humans, and rightly so. So actually, there was a study already conducted like this. It was a pilot study, and we are actually awaiting funding to proceed with a larger scale trial. But in this proof of concept study, we kind of replicated the experiment as in the mice. So we looked at individuals at the subacute back pain stage, and we treat them either with L-DOPA plus naproxen or placebo and naproxen. And what was found over 1 year was that the females that received L-DOPA plus naproxen showed very little pain 1 year later, as you can see here on these line here in pink. However, if they receive placebo, they actually do much worse. And I should mention that this effect was actually on this pilot study only observed for females only, and we do know that the dopaminergic system shows some sex-specific effects, so it could be the case that this treatment might be more efficacious for females rather than males, and this is again something that we are using to probe the system. So now I'm going to move on to the last part of this talk, where instead of talking about kind of the mesolimbic system and dopamine, I'm going to instead talk about memory and how memory can potentially affect pain. So I already hinted in the past that some of your pain experience is driven given your expectations, and your expectations may kind of prime you to perceive pain as more aversive or more intense or more salient. And this is something that is not very well described, but there's actually a very interesting case study in patient HM, which is actually a very famous neurological clinical case study, where basically this patient, because of intractable epilepsy, got his amygdala and hippocampus removed. So this patient could not form new memories. And most people study these subjects based on their ability to form memories and their affective state, but almost no one really looked at whether he could still feel pain. But there was one study that actually tried to test whether this patient HM, could feel pain despite not having a hippocampus and amygdala. And to quote the study, they say that HM did not label any stimuli painful, no matter how intense they were, nor did he withdraw the stimulus gun that they were using to stimulate the patient with. So basically, in other words, this patient that didn't have a hippocampus or an amygdala did not feel any pain, which is very surprising. And the way why we think that this is the case, going back to the model that I kind of discussed, is that we think that whenever your brain makes a decision about whether to transform a given sensory reality into pain, it also leans onto your past experiences that are stored in memory. And basically, your past experiences are also going to help shape a bit of your current pain experience. So this is actually a very interesting study where what they did is they looked at patients with chronic back pain, and they asked them, at this moment that you're coming to the lab, how much pain do you feel? And then over the phone, 36 weeks later, they actually asked the patient how much pain they remember feeling at the time that they came to the lab, so in the past. And what they found was that almost all patients tended to over-exaggerate their pain, which is perhaps not surprising, but also that this exaggeration of their pain was linked to structural properties of the hippocampus, which is, again, an area that is very important to form new memories and recall old memories. So in other words, these distortions of the hippocampus seem to be affecting the way that a patient can recall their own pain experiences. So in the same longitudinal study that I showed you before, where we were looking at the nuclear comments to the medial prefrontal cortex connectivity, it was also found that the volume of the hippocampus at the subacute stage could predict which patients would go on to develop chronic pain 1 year later, which is shown here. So basically, the subjects that had subacute back pain but then persisted had a much lower hippocampal volume compared to both patients that recovered from their pain and also healthy subjects. Again, linking the ability to kind of form new memories and the structural integrity of the hippocampus with the probability of developing chronic pain. And to take this step further, we actually just completed the study. So this study was actually published about 2 weeks ago, where we looked at a slightly different model. So far, we've been talking about low back pain. But here, what we did is we looked at whiplash injury. And whiplash injury is a very interesting model to study the transition from acute to chronic pain, because on one hand, there's a very high prevalence of chronic pain in patients suffering from whiplash injury. It's about 50% about 1 year later. But also, the whiplash injury usually is associated with a strong emotional component and a lot of formation of new memories, because this injury is usually associated with, for instance, a car crash that usually can even create post-traumatic stress disorders. And the other very interesting thing that we can do with whiplash injury that we cannot do with, for instance, subacute back pain, is that we know exactly when the patient had his accident. And so we know what the exact timeline of pain onset. And so what we did is in this study is that we collected a very large sample of about 249 patients that were scanned within 48 hours of their accident. So this was all motorized vehicle crashes. And then we followed their pain over 12 months. Now, we did lose a bunch of patients to follow-up, and in the end, we ended up with 111 patients. 53 of these patients had no pain at all 1 year later. 58 patients actually reported pain superior to 30 out of 100, so significant pain. And we, again, follow their pain over time. And as you can see here, the patients that 1 year later developed chronic pain, their pain remained relatively constant throughout the whole study. And the patients that recovered, their pain kind of went down over time, as you can see here. Now, these groups were pretty similar when it comes to their depression, their anxiety, and other parameters that we tried to assess. And what we found was that indeed at this very early acute stage, so within 3 days of the injury, both the hippocampus, which is again this area that is important to form new memories, and its network, so the areas of the brain where these memories are actually consolidated in, were incredibly upregulated in the patients that went on to develop chronic pain compared to the patients that recovered. And it got even more interesting. So what we also looked at was whether the time that has elapsed since the injury to the time that we actually scanned this patient would show differences in the hippocampal activity. And what we found was that 1 day after the injury, the two groups, the patients that recovered or those that developed chronic pain, their connectivity was pretty much the same. But then 2 days after the injury, there was a little more connectivity dissociating these two groups. And then 3 days after the injury, these three groups are almost completely dissociable. So in other words, what we are finding here is a response that is evolving over time and seems to be causing chronic pain later on. Now, we also found in this group that this connectivity seems to be modulated by anxiety. So basically, this increased connectivity that is imparting risk for chronic pain seems to be highest in the patients that also report high anxiety levels after their car crash. So in other words, what we now think is that there's usually a stressor. It creates these largely affective and emotional states in the brains of patients, which then allows the patient to form new maladaptive memories, which then impart risk for the development of chronic pain later on. Now, I want to briefly touch upon two preclinical studies that kind of followed this trend and that really tap into these causal mechanisms of whether learning and hippocampal plasticity can induce risk for chronic pain. So this study, published by Abkarin et al. In 2016, what they did is they stopped the ability of the hippocampus to generate new neurons. So the hippocampus is a very unique structure because it is the only structure in the brain, as far as we are aware, that is able to create new neurons. And it is thought that the creation of these new neurons is associated with the ability to consolidate new memories. So we can actually use genetic techniques to stop this process altogether, so to stop and block neurogenesis, so the formation of new neurons. And what was found on this study is that in these mice, again, these are mice with spare nerve injury, so you cut their sciatic nerve, 2 out of 3 branches of the sciatic nerve, and you poke these mice with these filaments to see what's the force that you need to make the mice withdraw their paw. And what was found here is that in mice that had perfectly normal neurogenesis, so they were able to form new neurons, we call them wild type, but that had a spare nerve injury, they show a lot of mechanical allodynia. So basically, they become very sensitive to pain. However, if you block neurogenesis, again, genetically, then you will see that these mice end up never developing mechanical allodynia. So by blocking the ability of the brain to form new memories, you are also able to block mechanical allodynia. And this was also shown through a brain stimulation study that was performed afterwards, where now, instead of blocking neurogenesis, what was done was to inject glutamate, which is excitatory, in the hippocampus in these mice that had, again, a spare nerve injury. And what was found is that when you stimulate the hippocampus, you can completely revert this mechanical allodynia temporarily. And this was even repeated twice. So what happened here was that the mice suffered an SNI or sham injury. And basically here there was an infusion of glutamate. And as you can see, there is like a complete reversal of mechanical allodynia for the mice that actually got infused with glutamate. Then after a couple of days, it goes back to normal, and then you infuse it again, and you can again revert mechanical allodynia. So it seems like stimulating the hippocampus is able to revert this potential pain experience in these animals. So I want to end by kind of saying that one of the things that we are doing now, not just us, but also a lot of other researchers, is to try to get some of these mechanisms and try to actually causally manipulate them, but in humans. So one of the ways that we can do that is using, for instance, transcranial magnetic stimulation, where basically we apply magnetic pulses to a given brain area, and we are able to either excite that brain region or inhibit that brain region. And this is a technique that is actually used for depression. It has been FDA approved for depression. And now we're trying to understand if, for instance, if we are able to inhibit some of these regions that are forming new memories, are we able to prevent the transition from acute to chronic pain? I also want to say that we don't necessarily need to employ these more invasive or non-invasive brain stimulation techniques. There has actually been quite a lot of success at trying to change these memories using, for instance, psychotherapy. So this is actually a very recent example that had very significant results. So what they have done in this study was they recruited patients with chronic back pain, and they were split into 3 groups. Either they received a psychological treatment that I'm going to describe in a little bit, or they received placebo, or they received their usual care. And again, this psychological intervention focused on 3 key aspects. So the first one is called neuroeducation. So basically what you do is you teach patients about the things that we've been discussing over the last hour. You teach them that their brain plays a big role in forming their own pain experience. You teach them that the brain can either downplay or upregulate nociceptive input arriving to the brain. And so when patients learn that their brain plays an active role in their pain experience, they are able to try to change some of these beliefs, and as a consequence, they might have different expectations about a given treatment. Another thing that is typically done is reconditioning, where you basically work with the subjects so that he experiences some movements and some pain sensation to understand that this is a normal situation rather than being these hyper-arousal, very worrying states. And by reducing patients' anxiety, for instance, to perform movements, basically what you are doing is you're reconditioning your brain to stop associating a given movement with this heightened pain state. And this might in turn reduce the effect of your brain over-interpreting your nociception as being incredibly painful. There's also a third component to these psychological interventions that try to regulate the emotions of the subjects, which again, we also know that impacts the way that the brain perceives nociception. So what they found, as you can see here on the left, is that the individuals that received this therapy called pain reprocessing therapy actually showed very substantial pain relief compared to patients that either received placebo or throughout their usual care, and this pain relief was actually sustained over a year. So this is actually very significant results and much higher than most therapies that we are actually providing our patients with. You can actually see here that there's a very significant number of subjects that had a complete reversal of their pain. So basically they recovered completely. They reported no pain after this with no peripheral treatment, just basically by changing the beliefs of the patient in how their body is sending signals to the brain. So I want to very quickly conclude by saying that pain, like we discussed, is a very complex, multidimensional experience. It requires not only nociception, but also an active interpretation of the brain of these nociceptive inputs. And so pain is obviously influenced by properties of the brain, is in fact generated by the brain itself. Now, we have discussed a little bit about the mesolimbic system, and I have shown you that these areas, including the nuclear accumbens and the ventromedial prefrontal cortex, seem to be causally involved in the transition to chronic pain. We know in humans, including, that if we are able to treat these patients acutely with, for instance, L-DOPA and naproxen, we might be able to block the progression of chronic pain. And obviously, it's much easier to prevent that rather than to treat. So we obviously need a lot more studies, but it seems to be quite promising to try to study how we can tap into the system and possibly revert the chronic pain state. And in the end, I kind of briefly talked about how cognitions and memories also play a major role in chronic pain. And we are now starting to use both human research and also basic research with animals to try to address and narrow down some of these mechanisms so that we can both develop targeted tools, both pharmacological in terms of neuromodulation, but also in terms of, for instance, psychotherapy, so that we can at least reduce some of the burden of these patients. So thank you very much for your attention. Again, this is my email, and I'm happy to answer any questions that you have or any curiosities. So feel free to reach out. Thank you very much, and I hope this was useful. Thank you.